Okay, welcome back. I'll remind you that we are recording lectures in pathophysiology. This is very first chapter about cell and different tissue, and this is the second video in this first chapter. This video we will speak about mitosis and meiosis, and it will speak about different organelles. So what about mitosis and meiosis? These two processes, by the help of these two processes, cells are divided. But mitosis and meiosis, and they are of course different processes. Mitosis take place in almost all the cells. By the help of mitosis, cells are divided. And the consequence of the mitosis is that we get two daughter cells which contains same amount of the chromosomes as the mother cell. For example, if the mother cells in the human case, normal cell contains, 46 chromosomes, 23 pair of chromosomes. After division, after mitosis, two daughter cells will contain also 46 chromosomes and 23 pair chromosomes. But when it comes to meiosis, by the help of the meiosis, division takes place, but amount of the chromosomes is divided. For example, and the meiosis takes place in the germ cells, in the spermatocytes, development of spermatocytes and oocytes. Why does it take place? Because spermatocytes and oocytes are then supposed to fertilize, to make one egg, one cell. So it's more practical to have the divided amount of the chromosomes, because they will make one and the one will contain normal amount of cells, because otherwise it will cause a very strange situation that in every division, in every replication, amount of the and in the fertilization, amount of the chromosomes will be increased. Okay, after the meiosis, the amount of the chromosomes in the daughter cells is 23. And they are not pairs, just 23 chromosomes. Okay, so mitosis in human cells reproduction is a complex process called mitosis. Mitosis contains 4 phases. It's prophase, metaphase, anaphase, and telophase. Before and after of the mitosis, there is interphase. This is a resting process where the amount of the organelles is produced. DNA, deoxyribonucleic is replicated, and so. Let's discuss all these phases. What's happening in each phase is very important. So in prophase, this is the very first phase. During prophase, the chromosomes became shorter, fatter, and visible. And the chromosomes have 2 chromatids, so 2 identical chromosomes itself, so 2 chromatids, which is the consequence of the replication of DNA during interphase. You see here what's happening. So chromosomes became more visible, and the nucleus and nuclear membrane disappear. Nucleus and nucleus membrane disappear, leaving the chromosomes in the cytoplasm. So you see here, centromeres, chromosomes, nucleus is disappeared here. You see that? Yeah. So this is what prophase happens. In metaphase, during metaphase, the 24 chromosomes, to each of the 26 chromosomes, each consisting of 2 chromatids, became attached on the spindle fibers. They became attached on the spindle fibers. This is what's happening in metaphase. So now what about in anaphase? What's happening in anaphase? In anaphase, the chromatids in each chromosomes are separated. So chromatids in each chromosomes. This is very crucial, very important. Chromatids are separated, right? And one chromosome from each chromosome then moves toward each pole of the spindle. So chromatids are separated. You see, though they used to be one chromosome, now they are separated. And what's happening in telophase, right? In telophase, we will get finally two daughter cells, and cell cleavage takes place. Two chromosomes, two daughter cells are produced, nucleus is reformed, and we will get two daughter cells, which contains also 46 chromosomes, because only chromatids was detached, but amount of the chromosomes is same. This is very important, guys. Please pay attention. A cell cycle, cell cycle is the whole cycle. And mitosis is only very little part of the cell cycle. For example, if you say the cell cycle is 24-hour, mitosis is only 1 hour. So here is the figure of the cell cycle. You see this is a mitotic phase, but what's happening before the mitosis? There's interphase, as we already mentioned, interphase, different process takes place in G, and there's G2 phase, S phase, and G2 phase. But not all the cells are dividing all time. And some cells does not divide, right? For example, neurons does not divide that often, and so many cells don't divide that often. Therefore, they are not going this mitosis phase, right? And all these cycles the time. Therefore, they are found in G0 phase, exists from cell cycle, non-dividing cell. Yeah, but dividing cells goes to this cycle all the time. For example, what's happening in interphase G1 phase, which will be 8 to 10 hours. Cell metabolically is active during this phase, duplicates organelles and cytosolic components, centrosome replication begins. S phase, DNA is replicated. G2 phase, cell growth continues. Enzymes and other proteins are synthesized. Centrosomes replicated, completed. And mitotic, we already mentioned, and you see the final consequence. Okay, so now next thing, meiosis. We already mentioned what's meiosis. The consequences to we have the daughter cells, which contains only half of the chromosomes. Meiosis has two stages, first meiotic stage and second. So there's two divisions. Each stage contains prophase, metaphase, anaphase, telophase. Let's go quickly, because this is, guys, just a reminder you are supposed to know all this information. It's just reminding you. So first meiotic stage, but of course we will, I know that maybe sometimes will be difficult, you may have questions, please write down all these questions, and during online classes we'll speak about this. So first meiotic stage, what's happening in first meiotic stage? Each has prophase 1, metaphase, anaphase, and telophase. So prophase 1 in first meiotic stage is similar to the prophase in mitosis. However, instead of being scattered randomly, the chromosomes are arranged in 23 pairs. For example, 2 chromosome numbers, 1 will pair up. So chromosomes are arranged in pairs. That was not happening in mitosis. So why is it important? Because during metaphase and anaphase, in meiosis, not chromatids are separated in first meiosis, but pair chromosomes are separated. In first meiosis, guys, please pay attention. That's what's happening. So chromosomes are separated, pair chromosomes. One pair will end up in one cell, another pair will end up in a cell, not chromatids. Each chromosome will contain chromatid. So after the first meiotic stage, we will have 2 daughter cells which contain 23 chromosomes, but each chromosome will have 2 chromatids. Okay, I'm not going to do this. And what happened in second meiotic stage? very important thing also, before the second meiotic stage, there is not interface, there is not DNA replication. They are going directly to second meiotic stage. Why is it that important? Because in second meiotic stage, the cells produced by the first meiotic division now divide again, and now chromatids are separated in each chromosome. And we will end up then two daughter cells which contain 23 chromosomes, but each chromosome does not contain chromatids, right? This is very, very crucial, very important actually. And fusion of gametes, when the gametes each with 23 chromosomes fuse together, a zygote is formed, you know. One chromosome each pair comes from the mother and one from father, we know this. Okay, now let's start speaking about organelles. Like human body has organs. Cells are the organelles. All cells contain many organelles. You see here all the organelles. Let's go quickly on these organelles. The first one is reticulum, endoplasmic reticulum. Endoplasmic reticulum is a place where lots of things are done. Carbohydrates in our storage, different proteins are synthesized. There are two types of endoplasmic reticulum. This is granular or rough endoplasmic reticulum, which means that ribosomes are found on the surface in this, and therefore their protein synthesis takes place. But next one is the agranular, smooth endoplasmic reticulum, where the ribosomes are not found. Free of ribosomes, you see, this is smooth, there is no ribosomes, and this is a rough endoplasmic reticulum. You see the ribosome here. So the next one is the Golgi apparatus. The function of the Golgi apparatus is the collection of this contains a collection of the different tubes. Function is to produce and storage different enzymes, for example, enzymes for the lysosomes, to produce different secretory products from the cells, and so on. So you see here, this is Golgi complex, Golgi apparatus. Golgi apparatus, very important. What about lysosomes? Lysosomes is also very important organelle. Organelles bound to the membrane it contains different enzymes. And for example, so in phagocytosis, when for example, different bacteria, phagocytes, they ended up and they are dead in the lysosomes inside of the cell. So digestion of material taken up by endocytosis, phagocytic organs, breakdown from cell components is a function also. In normal cells, why this is very important and funny. So breakdown of cell components during embryological development. The fingers and toes are webbed. The cells between the toes and fingers are removed by lysosomal enzymes. After baby's birth, the uterus, which weighs around 22 kilograms at full term, is invaded by phagocytic cells that are rich in lysosomes. This reduces the uterus to its non-pregnant weight, about 50 grams within about 9 days. Yeah, so it contributes to hormone production and so and so. The next organelle is a peroxisome. Peroxisome is quite similar to the lysosomes, but are much smaller. They are particularly abundant in liver cells. They contain several enzymes that are toxic to body cells. Next one is a mitochondria. Mitochondria or mitochondrion single. This is a powerhouse because the main function of the mitochondria to produce energy, TCA cycle, tricarboxylate cycle, takes place. And there is also enzymes for fatty acid oxidation. The mitochondria has a 3 membrane. It's very, very important organelle. Let's see the picture. Here the mitochondria. Those are lysosomes. Cytoskeleton. The cytoskeleton is a lattice-like collection of fibers and fine tubules in the cytoplasm, and it's involved in the cell's maintaining and alteration of its shape as required. They are microfilaments, microtubules, intermediate filaments. So microfilaments are the rod-like structures, 6 nanometer in diameter, consisting of protein called actin. Mast both actin and myosin are found. Yeah, microfilaments. Microtubules are relatively straight, slender, cylindrical structures that range in diameter for 18 to 30 nanometers. They consist of protein called tubulin. And intermediate filaments. The intermediate filaments range in diameter from 8 to 12 nanometer, as they also help to determine the shape of the cell. Examples of intermediate filaments are neurofilaments found in nerve. Actually, that was the last topic in this second video. We will record third video, and that will be enough. So thank you guys for watching. Next video, we will speak about type of cells and different types of tissues. See you next video.